biochemical properties that actually cause the protein to fold up into some structure, and the structure goes on to attain certain function. Now, some function can be clearly defined with a specific structural configuration. So next slide. And so this is specifically in the case of, say, enzymes where its function is specifically determined by the active site residues. And so how they oriented against each other's, and that gives you, like, say, catalyzes some whatever transition state. And this is also the case for certain protein-protein interactions where there's a specific epitope that a given protein binds onto. And if that epitope isn't there, then you can't bind onto that thing. And so these are what we call structural motifs. And so if you had a generative model that is amenable to structure guidance, you'd be able to access this problem class. But there are some functions which are not dependent on the structure. Next slide. Next. Oh yeah. And so these functional motifs are just sequence dependent. And in fact, if they have a predefined structure, that would limit their functionality. And so, for instance, if you had a generative model that is amenable to sequence together design, then you can go ahead and access this problem space. But there's also other functions that are specifically only arise with the interplay between sequence and structure. And so a good example of that is multistate design. Next slide. And so within multi-state design, a given sequence has a specific structural state dependent on some conditions, but then in another condition, they have a different state. And so in order to access to this problem space, you need a generative model that can get guidance from both sequence and structure. And so an ideal generative protein design method would be able to access to all of these three problem space. So you want a method that can do both sequence and structure. Next slide. And now typically what the literature has done and the field in general, they've generally separated protein design to two distinct steps. So first you define a given objective, and this objective is usually determined by some criteria which you determine that function is. In the olden days, this was specifically they have some structural attributes. And so this structural attribute, you want your given protein to have this. So you first define this, and then you go ahead and find a backbone that holds that given structure. And then from there, you go ahead and use another model to go ahead and thread on or design a sequence onto that backbone, and then you get your sequence output. Now, this paradigm really hasn't changed from the old days with like Rosetta-based design. Even new age deep learning methods, such as diffusion, still rely on first getting that structure and then going ahead and using an inverse folding model to get your sequence out. But more recently, language models have actually been able to skip the structural part, where you can go ahead and define a function and then get a sequence that directly corresponds to that. Next slide. Now, what we sought is to go past these previous methods and give you something that gives your protein, something which we term ProteinGenerator. And what ProteinGenerator does, it gives you directly the structure and also the sequence out. And so this is ideally what you want. And then next slide. And so how do we actually get there? So how we actually got there is by using this structure prediction network called RoseTTAFold and using that as the backbone of our generative model. And so the reason we use RoseTTAFold is RoseTTAFold can actually take in both sequence and structural inputs and actually output sequence and structure out. So what that allows us to do is to guide for specific structural and sequence attributes by directly giving those to the model. And then once we get that, then we both get the sequence and structure. Now, previously, people in the lab have also developed structure diffusion with RoseTTAFold, but what we do differently is we do our diffusion within sequence space. Next slide. Now, the question is, why did we choose to go through to use sequence diffusion? And so, next slide. Well, the reason why is because sequence is actually has really nice properties. For one, if we have a diffusion model within sequence space, that allows us to use guidance within sequence space. So mind you, say we have some activity function data, we can go ahead and train a classifier on that activity, on that and that sequence of activity data and actually guide the generative process toward those active regions within sequence space. Now, the reason why this type of data is more plentiful is just because it's way more cheaper to sequence proteins rather than actually get the structural out. And so we now have access to this specific type of data to which we can guide the model towards. But then even more so, there's certain attributes of a protein that are specifically dependent on the sequence, not on the structure. So in this case, say you're trying to develop a drug, certain developability properties directly arise from the sequence composition within that given within that given drug, rather than the actual specific structural arrangements. And so, if we have our generative model within the sequence space, then we can go ahead and guide towards those developable or desirable attributes. And then another big important part is structural actually arises from sequence. So we can still guide for specific structural attributes just from sequence space alone. Next slide. Okay, so now that we've motivated why we want to do diffusion and sequence space, now the question is, how do we actually represent our how do we represent sequences in something that the model would understand, specifically within this diffusion paradigm? Well, we chose to actually represent our sequence in terms of one-hot tensors. And with this representation, we can simply, as the normal continuous diffusion literature is done, we can just add Gaussian noise to this and then slowly remove it within the typical diffusion paradigm. And now what this also allows us to do is we can use the very same classifier-based guidance formulation that was popularized within diffusion literature without having to make our own. And so this is really nice because then we can have this very simple, easy to implement thing, and we can use all the previous work people have done within generative work to guide us towards where we want to be. And so here we have a representation of that. So what we give the model is not only this one hot tensor, but we also flag on a residue-wise basis whether that region is being diffused or not diffused. So in the grayscale, those regions are not being diffused because then we also pass in we pass that information to the model, so it doesn't need to diffuse there. But then the region within purple here, the model is allowed to do whatever it wants to get us to where we want to be. And so this is kind of like, if you look up, is the sequence logo representation of what different amino acids are being decided upon within the diffuse region. Okay, next slide. And so this formulation actually has appeared previously in the literature, specifically in this analog bits paper, where here they represent, they actually go ahead and represent an image in discretized form, specifically in this bitwise representation. And so it's really the same formulation here. And then next slide. And another cool thing they did in this paper is they came up with this concept of self-conditioning, whereas essentially, rather than having the model only reason with xt as what's done here, we also, in addition, having the model reason with xt to give you x naught. In addition, we also provide the model what its previous prediction of x naught is here, and when it's predicting the next x naught, in addition to what xt is there. And when we found this really fine, we find that this stabilizes training and also helps us converge faster. Okay, next slide. And so putting all together, how this looks when we're actually generating is we start off with a completely noise sequence, so in the left here, and then we task the model with continuously denoising. And so here at the next time step, we see that it's a little bit denoised, but then one thing to know is we're also applying losses on the structure, so we also get the structure out as well. And so here we have a joint sequence structure paradigm. And so by continuously doing this, we get something that more and more resembles an actual protein, both at the sequence level and at the structural level. And then if we zoom in what's happening in the model, we can actually, next slide, please. We can actually start seeing how we can go about doing classifier guidance. So in this case, each time the model does a single step, we predict X naught. And once we have that prediction of X naught, we can actually take gradients from another classifier with some attribute we care about and actually add it onto that predicted X naught, noise it, and then pass it back to the model again. And via this way, we can guide towards any attribute we care about. Next slide. Okay, and then here we'll let Jake take it from.
Yeah, thanks, Sidney. My name's Jake. I am I'm my fourth year in David's lab. And yeah, I guess kind of like Sydney mentioned, like we were sort of inspired by like what the previous generation of people who had built some of these generative models did. And that's mostly just like really sticking true to the structure prediction architecture and looking at this process as more of like a fine-tuning process than like training a generative model from scratch. And so in that vein, this schematic sort of goes through like how we think about the model in the context of like this sequence diffusion process. And so if we start with like a protein sequence, which is some like real ground truth sequence, we featureize this, like Sydney was kind of talking about, according to this like analog bits formulation, where we end up with essentially sort of like a latent representation of the sequence that is continuous, and this allows us to then be able to add noise to this tensor directly. And so really this forward diffusion process is just adding sampling Gaussian noise around this initially like discrete looking object. And then really what we use RoseTTAFold for, and what RoseTTAFold is like really good at, is it's a nice architecture that is able to mix these different types of conditioning information. So this is the 1D track mostly carries the sequence. The 2D track is containing this pairwise information with the structure. And the 3D track is carrying the explicit 3D coordinates. And so after noising our sequence to some T, we are able to provide motif conditioning information from the sequence. And we'll talk about how we do this in a second. But some of these features, including like DSSP information to predict the secondary structure, are all concatenated onto this 1D track. And then in the 3D track, we can provide things like the active site for a protein, if it's an enzyme that we're scaffolding, or some other structural motif as well. This conditioning information can be fed in through 3D track here. And then kind of like Sydney mentioned, like this work that we've done is certainly somewhat of a departure, I guess, from like the normal diffusion literature, in that like we're looking at this as more of like a fine-tuning process, where we are, first of all, we're starting with a pre-trained version of RoseTTAFold.
Where we have the package ready to go.
Oh, hello. Oh, maybe it's someone else. Okay. Yeah, I guess like I was saying, we see this more as like a fine-tuning task. So we're actually applying the same losses that if you look in like the RoseTTAFold 2 paper, they apply over the structure. And then over the sequence, we apply a CCE loss, and we'll kind of talk about some other specific losses that we ended up using here as well. In terms of choosing a noise schedule, like this was quite important for our task. Doing this in discrete space, it becomes easy for the model just to be able to argmax the sequence, especially at low time steps, depending on what kind of schedule you use. And so actually this came from this work doing this like language model, this like LM diffusion, where they showed that the square root formulation of the schedule, where if you space these alphas along a square root schedule, basically, when you visualize the standard deviation that you'll be using to sample the noise with, you can see that this square root schedule scales quite quickly. And basically what that means is like you are corrupting the ground truth tensor with like more noise earlier on, which sort of washes out any signal earlier from like the one hot encoding that we're initially featurizing with. And so yeah, we kind of found that the square root schedule worked best for us, and sort of, yeah, the advice of like these people from the LM diffusion, we went forward with that. I think in terms of the tasks that we're doing during training, this was really important and took a while to kind of figure out like what tasks were most important to be able to sample good proteins. One of the most important things was really balancing the tasks between doing like the standard diffusion denoising objective to structure prediction and backbone design. And so really what we do here is like during the diffusion task, we just noise the sequence like we normally would, and we don't provide any kind of structure. And the model is always tasked with denoising the sequence, but also producing the corresponding structure as well. For the structure prediction task, what we do is we provide the sequence denoised and no structure. So basically, like at time step 0, we just show like the denoised sequence and ask the model to recapitulate the structure. And And then for backbone design, what we do is we noise the sequence to big T, and we then just provide the structure as conditioning information, with the goal that the model will then predict the sequence that corresponds to that input structure that's conditioned on. From a masking perspective, like we sort of implemented two different styles of masking, like the sparse and these continuous masks, where these contiguous masks sort of are akin to something that you would do for like motif scaffolding, where you have a contiguous segment either in sequence or structure space, and you would like to condition on that. Or for active site scaffolding,
Like that. That's a good point. Yeah.
Because again, it's like based on my understanding of.
Okay. For the active site scaffolding, where the residues are further away from each other in sequence space, we use this like sparse masking, where we just randomly sample positions along the sequence to unmask and condition on. And then we also trained some of these models with this DSSP conditioning, where we append this information to the 1D track that tells you basically, is this for each residue, is it what type of secondary structure it is, including a mass token as well. In terms of the data that we trained this model on, it's all coming from the PDB. I forget the exact cutoff date, but they're using the same training splits that RoseTTAFold 2 uses as well. And so, yeah, I guess just to look at a little more like how this information is getting fed into this is borrowed from the RoseTTAFold 2 paper, but right in this 1D track is where we're actually providing the sequence. And here, like in these design models, we're never providing an MSA. So we still maintain the MSA track, but we just use a single sequence. But we can append to this the time step information. So, like what diffusion step it is, some other kinds of masking information, as well as the secondary structure features as well. In the 3D track, we featureize this with like in the 2D track with the structure. So, we use this like SE3 equivariant transformer to encode the raw 3D coordinates, and then the pairwise distances are encoded as well for whatever template like structural motif we would like to provide the model. I guess to put this like a little bit more concretely, looking at like how this sampling, how the training process actually works is, right, we one hot encode the sequence, and we apply some transformation to shift this to the analog bits formulation of -1, +1. Then we sample T uniformly. We, because we're doing self-conditioning, we both generate XT and XT plus 1. And then without gradients, we do this pass through the model to generate some self-conditioning information. And we do this about 50% of the time, like they suggest in the paper. And then we use that self-conditioning information to then predict, we're always predicting these ground truth X naught prediction. And sort of the three losses that we're using is the CCE loss over the sequence, which in the analog bits paper, they actually do suggest and mention that you can use either MSE or CCE loss. We use this KL reconstruction loss, which this actually was suggested to us by these authors from the LM diffusion paper. I don't know if I guess I have a good intuition for like why this is absolutely necessary, but empirically it does seem to help when we use this loss. And here we're basically interpolating our predicted output to XT minus 1, and then we do the same interpolation for the ground truth, and then basically apply a KL loss over those reconstructed XT minus 1. And then also we maintain the same structure losses that were used during RoseTTAFold 2 because we want to ensure that structure prediction capabilities are still there. And the model is able to realize like real structures. Come sampling time, really is pretty pretty straightforward. We sample some noise. We pass this noise through the model with time conditioning and whatever other conditioning information we have. Kind of like Sidney mentioned before, like this is where like if we want to do motif scaffolding, we'll provide like a mass token for those residues which are already fixed. And then like Sidney mentioned as well, we can also apply gradients from a classifier or from a regressive model that we train to do guidance. Our formulation for XT does actually not do this interpolation between X naught and to get XT minus 1, we're actually not interpolating from X naught to XT. We do this stochastic formulation, which LM diffusion also suggested. And then at the end of this, the idea is that we then just argmax in the sequence after we've taken we've gone through the full trajectory. And so, yeah, this is just like an ablation to show that like, kind of like I just mentioned, like actually conditioning on XT during the reverse process actually seems to hurt the network more than it helps. And so we actually went for just using not for the interpolation formula, but just the stochastic formulation, similar to how the forward process works. And so that led us to look to better sampling, as you can see here across these different lengths. And then looking at the sequence profiles from inference compared to natives, we can see that some of the distribution seems to match, although certain residues are certainly oversampled in our model. I think like in looking at this, and especially in comparison to MPNN, is kind of interesting. I don't think that this plot is specifically as figures in the paper. But yeah, what you can kind of see is like there are some interesting residues that both like glutamate that both MPNN and this protein generator model seem to both favor for some reasons. And then on the left here, actually, this figure is, so if you take these backbones from native PDB and you redesign them with either MPNN or with the sequence diffusion model, in this case, for our model, we're basically providing the backbone as conditioning information, very similar to this like backbone design task, and then we're fully noising the sequence. And so really the diffusion process is generating the sequence condition on this backbone. And then here, what we're showing is you fold actually these sequences with MPNN, or sorry, with AlphaFold. And what we can see is that like the structural quality after generating the sequence from these models, whether this MPNN or from our protein generator model, are able to find sort of better sequences that fold up into this structure than those in the PDB. So really in single sequence mode, very few members from the PDB are actually able to fold correctly. And so this is all with single sequence mode and AlphaFold. You see, we're not really maybe quite doing as well as MPNN is, but we're able to sort of shift this distribution certainly towards towards like higher structural quality for the sequences that we're generating. I think one of the big questions we often get asked is like, okay, how does this compare to RF diffusion? And so I think that it's clear to see that like in some tasks, like we're not doing as well as RF diffusion. I think that especially for this like motif, this is for a motif scaffolding task here on the right. We can see the success rate for a method like RF diffusion is certainly higher. And we really tried, I guess, for most of the paper to sort of find these specific applications where you want to do like sequence-based guidance to emphasize like the interesting use case, I think, for this kind of model is when you want to be able to do some kind of a guidance in sequence space. That's really not approachable when you're doing diffusion over structure space. Here is a comparison to another model for sequence diffusion, which is this EvoDiff model from the people at Microsoft. And I think what's interesting to compare against these two is that, right, like in Protein Generator, we have this whole structure prediction part of the network, and also we're able to input explicitly structure coordinates as well. And so really, I think that's really why we see this boost in performance over EvoDiff, is that when the model is able to condition on the structure, the sequences that come out, and when you fold them, are usually higher PLDT. And interestingly enough as well, is that if you look at the ESM perplexity distribution of the sequences, they actually seem to match with the distribution from UniProt quite well. Whereas if you were to just randomly sample amino acids, this is sort of what the ESM perplexity would look like. And then EvoDiff is in the middle here too. And so now, I'm going to pass it over to Sam to kind of go through some applications of the model.
Cool, thanks. So kind of continuing from what Jake was saying about, there are certain use cases where structure diffusion models like RF diffusion really excel, and where sequence diffusion models like Protein Generator excel. And there are some overlap, and there are some areas without overlap. So this is kind of where we wanted to, design some proteins, test them out in the wet lab, and see if this sequence guidance for specific design goals was really going to work out as we hoped. So we tackled 4 different design goals in the paper, where first we wanted to enrich for rare amino acids that were underrepresented in inference time for Protein Generator. We designed symmetric and repeat proteins. we scaffolded functional sequences into proteins, and then we also did multi-state design with ProteinGenerator. So we'll just kind of walk through the experimental data for these 4 design goals. And so starting off with the amino acid biased sequence guidance, what we're doing at each step is given some XT and some amino acid that you want to upweight or increase the percentage of in your final output protein, you first select the top K positions in your continuous sequence representation with the highest probability at that time step for that amino acid. And then you apply some sort of scaling factor to those positions. And you repeat this process for the entire diffusion trajectory, which is a little bit more complicated than just, upweighting tryptophan, for example, for every single residue. And the reason we weren't able to use that naive implementation is because it led to a form of mode collapse where the entire sequence just ended up being tryptophan. So we have to do this sort of process where we're selecting at each step the top K positions that the model thinks are going to be tryptophan and then upweighting those. And so what we found is that when we're taking these rare amino acids that are, both underrepresented in our inference experiments and the PDB, such as valine, tryptophan, cysteine, histidine, and methionine, when we apply this amino acid bias targeting 20% of the protein being made up of it, we're able to approach that target value, which really suggests that ProteinGenerator is able to reason with these out of distribution proteins, since they're not really seen or understood during training because of the noise schedule. These underrepresented amino acids just kind of get lost. Like, this information is underrepresented pretty significantly. And when we look at designs generated using this process, this amino acid bias, we can see that they are quite different from native proteins. So it's not just sampling the few examples that it's seen during training. And this is just a multidimensional scaling plot of native versus proteins generated with this bias. So the first set of proteins that we took to the lab were these tryptophan biased proteins, which look pretty crazy with these giant bulky aromatic residues kind of packed inside and a couple of them sticking to the outside. And, one of the challenges with incorporating these amino acids, so tryptophan, for example, is that it would be really hydrophobic and greasy and likely to aggregate. And when we expressed these in E. coli and purified these proteins, when we looked at how they purified under size exclusion chromatography, both of these designs were extremely soluble and monodispersed, suggesting they're not aggregating at all. And an interesting spectroscopic property of tryptophan is its absorbance at UV 280. So you can see both for 50 milliliter cultures of an unconditional protein in gray versus the tryptophan protein in purple, you get about 5 times as much absorbance on the UV spectrometer. So this could be used as like a protein tag for low expressing native proteins. And when we examined if they're folding by circular dichroism, we're seeing the 2 kind of standard alpha helical peaks for both of these designs. So this was kind of encouraging first data. So we wanted to keep designing proteins with, increasingly difficult amino acids to just kind of jam in there. So we designed proteins with an increased cysteine bias, and sort of the interesting design challenge for packing a bunch of cysteines into a protein is that ProteinGenerator now has to reason over structure space in addition to sequence space. Because if a cysteine isn't paired and forming a disulfide bond, it's likely to be forming disulfide bonds with neighboring cysteine proteins, and which causes all sorts of problems with expression and aggregation and stability. So it's really critical for ProteinGenerator to be able to, see that there are other cysteines in the protein and design a sequence and structure that will form these disulfide bonds. So when we purified and expressed 2 of these designs, again, under size exclusion chromatography, they're soluble and monodispersed, suggesting they're not aggregating and they're monomeric. And when we looked at how stable they are by circular dichroism, again, we're seeing these alpha helical peaks, suggesting they're folding into the secondary structure that ProteinGenerator predicted. And when we heat the sample up to 95 Celsius, we don't see any significant shift in the CD spectra from the 25 degrees C spectra, suggesting that there's thermostable up to 95 C. But we wanted to confirm that these disulfide bonds were actually forming, which is a really critical part of designing these cysteine biased proteins. And when we took these 5 designs to mass spec, we ran them under mass spec in the presence of a reducing agent and the absence of a reducing agent, where the difference in this mass should indicate the formation or breakage of disulfide bonds, because the reducing agent will disrupt the formation of these disulfide bonds. And when we look at the difference in masses between all of these designs, we can see that almost all of them have exactly the same number of disulfide bonds formed as intended. So this was really encouraging to show that the model is also able to reason about structure without any structural prior. And just one last example is designing proteins with increased valine content. So valine increases the beta sheet propensity of sequence space that it's in. And so the AlphaFold models and design models recapitulate the increased beta sheet content we were expecting to see. And they look well folded and thermostable by circular dichroism. And when we take, a bunch of samples from the model, either unconditionally here on the left with no bias, we see that they're mostly all alpha helical with not really any beta sheet content. And when we add this valine bias, we see a significant increase in beta sheet content, suggesting that, again, it's able to reason about these structural properties that arise from sequence. So the next design goal that we tackled and implemented in ProteinGenerator was designing symmetric or repeat proteins, where given some xt in your diffusion trajectory and some predicted x naught from ProteinGenerator, after you add noise, you can symmetrize your continuous sequence representation to generate a symmetric or repeat protein. So in the case of this example, this is a 9-residue protein. You can take the first 3 residues of noise and symmetrize it across the other 2. And when we expressed and characterized these proteins, they looked good on size exclusion chromatography, which is this top row. Circular dichroism on this middle row, and for our thermal melts, they all looked stable up to 95C on this bottom row. And we were able to obtain the crystal structure of one of our repeat protein designs, and it was really great to see that even at the sidechain level of resolution, our design and AlphaFold models matched the crystal with atomic level accuracy. And so next, kind of building on these forms of sequence guidance, and we implemented sequence scaffolding using the masked tokens that Jake and Sidney have described. You can incorporate functional sequences into a de novo or designed protein just by specifying what the sequence is, and then ProteinGenerator will sort of fill in the blanks to generate a scaffold around those. And so some use cases that we were tackling with this were, for example, with endosomal escape. If you deliver a vaccine and you want it to escape the endosome to actually affect the inside of the cell, you need some sort of protein that will help your vaccine escape the endosome. So we wanted to design a conditional cage around membrane-lytic peptides. And so we chose this peptide called melittin, which is found in bee venom. And when melittin is, caged and inert, it can just kind of be inactive. But if melittin is released and allowed to interact with other melittin peptides, it forms this giant pore in the membrane of cells and lyses them. So this is associated with tumor cell death due to its membrane-lytic properties. So we sought to design a protein that had, a inducible cleavage site. So we chose a furin cleavage site directly flanked by our bioactive membrane lysing peptide melittin. And then we tasked ProteinGenerator with sort of filling in the blanks and designing a scaffold that will hold both of these sequences and in an inert form until we're ready to release it with our furin cleavage. And so while melittin is a flexible peptide, it's also well known that it has helical propensity. So we also incorporated DSSP and aka secondary structure conditioning to make the de novo scaffold alpha helical, as well as our peptide helical, and to scaffold the cleavage site in a loop such that it's sterically and flexibly accessible to the furin protease. And so this is the design model in gray and AlphaFold model in color of one such design that we expressed and characterized, where our furin cleavage site is in cyan and our melittin peptide is in pink. And when we express these in E. coli and purify them, again, they're soluble and monodispersed by size exclusion chromatography, suggesting it's not aggregating and is well behaved. And it looks very well folded in with alpha helical peaks by circular dichroism. And so now we have to test if this was actually a functional peptide cage. So we need to cleave off our melittin peptide at the furin cleavage site, release our melittin peptide, and see if we get conditional membrane lysis. And so this was a red blood cell lysis assay, which we used because when red blood cells are lysed, they release heme, which absorbs at 450 nanometers. So if you get red blood cell lysis, your absorbance goes up. On the left here are two positive controls, Triton X100, which is a strong detergent, and purified melittin that we just purchased from a company. And only when we incubate our design with furin protease do we see red blood cell lysis. So this was really exciting. And there are some people in the lab kind of carrying this work forward, which is also cool to see. And so sort of the last design goal that we tackled, which I think was the most challenging, was designing, well, one, implementing a multi-state framework in ProteinGenerator, and then designing one sequence that can adopt multiple folds. So sort of the setup for this design problem is, you know, to design one sequence that can adopt multiple folds, you have some full-length protein sequence at the top that has alpha helices and beta sheets, and after some cleavage event in the middle, splitting this into two separate proteins, you want both of them to the resulting, you know, children proteins, child A and child B, to be all alpha helical. So you need to find one sequence that can both adopt beta sheets and alpha helices. And so what this looks like kind of going into the model is you're running three different trajectories at the same time, where you're providing the model with both the full-length parent sequence and applying DSSP conditioning for helices and sheets. And then you're also providing the model with the two split children sequences with DSSP conditioning only for alpha helices. And the few use cases that like you might want to actually apply this framework for designing different protein systems are either for fold switching proteins, which might have different material properties where beta sheets are usually more stretchy and alpha helices are more compact and rigid. If you're like working with different nanomaterials, you might want to model a binder design problem in the apo and the hollow state, where you have a binder in the presence of your target and your binder alone to make sure that it's well behaved and well predicted in both states. Or you might want to design some sort of sensor or hinge system where you have a complex of two proteins that adopt one conformation, and you want your unbound sensors to adopt a different conformation. And so the way that we actually implemented this into ProteinGenerator is at some XT, you're providing the model again with these three different secondary structure conditioning strings, and as well as, you know, three continuous sequence representations for each of these three states. You're getting a predicted structure out from ProteinGenerator for all three of these states, and then we average the model logits across all three states to converge on one single sequence that's predicted with high confidence to adopt all three of these target folds. And so we designed a bunch of these computationally, and then we ran AlphaFold on all three states to predict the child A, the parent, and the child B state. And we were able to find good designs with high AlphaFold confidence and low RMSD to our design models, shown here on the top. So they're predicted to adopt these multiple folds. And when collaborating with the McShann lab at Georgia Tech, they were able to obtain a high-resolution NMR structure of the parent fold, indicating that we're able to design this complex alpha-beta fold. And the HSQC spectra of child A and child B compared to the parent show distinct regions of non-overlap. So like where there's pink but no black behind it, or purple but no black behind it, suggests regions of the protein where there are significant structural differences between the parent and the child. And so that we also took sort of like a zoomed out view of how well we designed these multiple folds, where in the HSQC spectra of each of these proteins, it's known in the literature that proteins with high beta sheet content sort of have their chemical shifts in this bottom left, zone of the HSQC spectra. So this is an all beta reference protein, whereas all alpha helical proteins sort of have this upright shift in their HSQC spectra. So when we take all of the parent designs, which are in stars, 3 out of the 4 of them are significantly shifted towards this bottom left quadrant. Actually, the interesting thing is this 4th design we did obtain a structure for, and it does match with alpha beta fold, which is cool. And all of the children designs are shifted towards this top right quadrant. So, between the AlphaFold models, the NMR structures, and these HSQC data suggests that we know that our parent designs adopt our alpha beta fold with atomic level accuracy, and our children designs are highly likely to be adopting this alpha helical fold.
Yeah. And so I guess one of the last things that we wanted to show, and we don't necessarily have like wet lab data to validate this, but using some data sets, we wanted to kind of take a stab at like simulating what an iterative design process might look like. And so you can imagine like a lot of people are interested in building this like lab in a loop kind of pipelines where you generate some experimental data, you can train some models on this data, and then basically use this data to then bias the next round of generated designs. And so using a pipeline like that, basically what we did was we took this data set. It's this like GB1 binding fitness data set where there are 4 positions which these people sort of explored all possible combinations of these 4 amino acids in this data set. And so kind of what we did is we tasked the model like masking these residues here in purple, asking the model to generate what residues it thought would fit in these positions, conditioned on the target as well. And basically, for round 0, we basically took a bunch of sequences, the first 96 sequences that the model generated, and then used that to initialize a Bayesian optimization trajectory, as well as we use this to also train classifiers on that we can then guide ProteinGenerator with. And so basically how this worked is like in round 0, both the BO and the PG formulations were both basically seeded with an initial set of designs that were made from the model. Once we gave the model these designs for BO, we're always training Gaussian processes at each step, and then using a UCB, upper confidence bound acquisition function, to sample the next set of 96 things to test. For ProteinGenerator, what we did was we trained these classifiers to basically discriminate between active and inactive samples, and then use the classifier gradients to guide the model during sampling time to sample the next set of proteins to test. And so in purple are actually the activities that if you basically use the data set to look up the activities of the sequences generated with the ProteinGenerator model versus with BO, we can see that consistently because ProteinGenerator like has this intuition about like sequence space. It's able to predict sequences that are more likely to be functional than just a standard BO process without any kind of like protein or understanding of protein prior, really. I think what's cool about the PG stuff as well is that we didn't really use any acquisition function to rank these sequences or anything like that. This is just the straight 96 first sequences that we sampled from the model. And these are the highest, the max fitnesses here in these bars, these dots. So now I'll hand it back over to Sidney to wrap things up.
That's perfect, taking Stan. So just in summary, I think we've showed that protein generator and more general sequence specific models or sequence and structure specific models can really expand the space which we can target with protein design. And so I think it's a really big benefit to the field. And here specifically, we showed the power of sequence guidance, where we showed that you can enrich for rare amino acid proteins. Also, it's super simple to go ahead and start generating repeat proteins. And if anyone is familiar with the literature back in the day on actually how to make repeat proteins, it was actually really complicated. So it's like super simple how like deep learning specific and sequence based makes this way more tractable. And then in addition to that, we've also shown sequence motif scaffolding, which can really give you access to a lot of functional space that does not necessitate structure, or structure might actually be harmful for that given function. And then stepping from there, we've also talked about how you can do sequence structure guidance to get some really complex logic going on within your protein design system. So whether that may be conditional peptide caging, as Sam has mentioned, but then also something really at the frontier of protein design is, can we actually design proteins that do not have a static structure, but can actually adopt distinct folds, which I really think is a super exciting research direction. So I'm excited to see whether this sequence whether the sequence first approach is really the way to go about doing this. And then on top of that, we also showed some work on activity guidance, which this is actually super exciting too, because as you can imagine, as people just get more as high throughput experimentation goes, we can directly just use that for sequence to activity data to really guide us to functional space, which is hard to define with sequence or structure because it's just so complex. But so again, thank you for taking the time to listen to us. And then next slide. And then just a big shout out for everyone who helped us with the paper, specifically some of our co-first authors who aren't here, specifically Jeremy and Lucas, and then also some other folks who were great too, Sam Mandel, Muna, and then the Andrew McShann lab at Georgia Tech for your NMR work, specifically Miriam and Claire, and then also everyone within the IPD who really made this research possible, our PI David, and then the administrative staff within the IPD, specifically Linda, Lukey, and Candace. So with that, are there any questions?
Thanks, Sidney, for answering all the questions in the chat box. Everyone have questions, you could just unmute yourself.
Not all at once.
We got a few more questions. The first is, can protein generator generate sequence condition on two structurally evolutionary related proteins?
Yeah, so it's a good question. So if the proteins look the same, then yes, because you can just provide one structure. But I guess in general, I'm assuming you were thinking of something a bit more complicated. Okay, so let's assume that they are structurally distinct. Then I think you can do the same formulation where you the same formulation we did within the multi-state design, where you just condition on each structure and then sum those and then give those as input to the next iteration. So I guess that's how I'd answer it. I guess Sam/Jake, would you see another formulation?
No, I think that sounds reasonable. Yes, there's another question about conditioning for GB1 activity. Did you include the IgG interaction or just the GB1 structure? I believe
just the GB1 structure.
Just the GB1 structure. Okay. I guess we just used the GB1 structure in this case, so we did not have the IgG the interaction.
I do think it's actually super interesting that we don't necessarily need that interaction because the sequence activity data set will hold that information. And so I think that's also still an interesting result, that you can still progress even without it explicitly.
Could you use to guide caging without a non-environmental trigger?
Yeah, so I think that's one of the interesting use cases for this is, well, it depends if your peptide caging needs to be covalent or not. So I'm assuming you're describing like sort of like a locker-like system where you have a peptide that's caged, and then in the presence of a protein that it'll bind to, it will uncage and interact with that protein. So I think it would be a pretty similar formulation, but you would also have to, you would both combine your sequence motif scaffolding, and you could also probably combine that with multi-state design, because you want to both model your inert peptide cage in the absence of your target, so you can ensure that your peptide is, not floppy or interacting with anything else. And then you also want to model it in the presence of your target to ensure that your peptide is predicted to be binding to your target in the presence of it.
Okay. We'll wait for one more minute for there's maybe some more other questions here. Oh, someone raised his hand.
Hi.
Oh, go ahead.
This is a really interesting set of work. I'm curious if you have any instances or if you've explored areas where you are conditioning on ambiguous sequence motifs. Maybe this is something that was addressed in the presentation in the paper and I just didn't catch it, but you mentioned conditioning on a single target functional sequence motif, and I think maybe your third case study, correct? Have you looked at instances where you have, multiple available potential sequence motifs that could fulfill the target criteria, and you're simultaneously conditioning on any one of those motifs as opposed to a single motif? I'm thinking kind of by analogy to your approach for conditioning on sequence composition, where you take sort of like a top K, and then I believe you're only guiding the model in the areas where it's closest to matching the target sequence. I'll pause there. That may have been a little bit of a word salad, but
Yeah, so we haven't explicitly done what you're describing, but something I've been working on that's sort of similar is, taking peptides that, one, don't really have any sort of known structure. These are, I'm working with peptides that get loaded onto MHC complexes. So they come from a totally different structure and then get processed into these like 9-mer fragments. And I'm designing mini proteins that scaffold 1, 2, or 3 copies of, any number of different peptides. So I have sort of like a library of like 10 different peptides I'm interested in incorporating into proteins. And I'm sort of just generating every combination of those 10 different peptides with either 1, 2, or 3 copies of them.
Okay, so maybe if I understood your answer correctly, you're exploring a context where you're enumerating out the set of potential conditional constraints you're attempting, and then you're running each one as its own trajectory.
Exactly. So it's not like choosing which one's best as you're describing, but it's in the same vein of incorporating different peptides using the model. Yeah.
I think to your question as well, like something we did explore, it didn't really make it into any of the experiments in the paper. But we also tried using like an MSA, like a PSSM matrix from like an MSA, and using that to bias the sequence generation process. We're able to find that at some scales that we were able to sort of generate sequences that were within distribution for the MSA or whatever the family is that you want to bias towards. So maybe that's a little more akin to what you're describing, with having a few different options for what your sequence composition could look like.
Yeah, closer, but obviously, as you're saying, because you're using an MSA, you have independent conditional probabilities at every position in the PSSM, when, of course, for other design targets, you might be interested in, a relatively complex sort of like undescribable joint probability that would be best modeled via like a nearest neighbor model or something along those lines. Does that make sense?
Sure. Yeah. That makes sense. Yeah. We did not explicitly explore something like that. Yeah.
Okay. Interesting. Very cool. Thank you. I could probably talk too long, so I'll mute myself now.
Thank you. Yeah. And thanks for Bo for inviting us here. And yeah.
Yeah.
Yeah. Thank you for your great talk, and I guess we could call it a day.
Cool. Thank you very much.
Thank you.